Hello student, today I am going to discuss about different instruments that is going to utilize in animal cell culture. Okay, so there are different glasswares. What are the different glasswares? Petri dish, culture flask, multi-well plates, cell scrapers. So culture flask, especially designed culture flask is going to utilize in animal cell culture. So you have to sterilize each and every glasswares that for animal cell culture in animal cell culture labs, and you have to keep your lab free from any viruses, any bacteria or any type of microbes. You know microbial cell, bacterial cell, plant cell and animal cell. But animal cell is highly sophisticated, delicate. So you have to maintain your animal cell properly. So you have to utilize uh, different laminar airflow. Laminar, that provide the aseptic condition. Tissue culture wood, that is laminars, where you can dispense your sample in the culture flask in aseptic environments. So what do you mean by aseptic? A sterile environment. Otherwise, your sample will be contaminated with viruses, bacteria, and your experiment will not be successful. So you have to dispense or transferring the media or inoculating the media with animal cell culture in highly sophisticated sterile condition inside the laminar. So this laminar provide the sterile environment and there is no chance of infections. So you have to take safety measure because the animal cell may be carrier of different dreadful viruses. You already know the coronaviruses. Okay, so all of you have to wear a mask, face shield, and keep your hands sterilized. Then HEPA filter, a special type of filter that is attached with laminar air hood. Okay, so that will provide a sterile condition when you are dispensing any animal cell in the animal cell culture tube inside the media. Okay, so there are some instrument which are going to utilized after inoculation of animal cell culture in animal cell culture flask. You have to put inside the incubator. Incubator, it is a device, it is going to utilize in animal cell culture media, multiplication of animal cell culture. So after inoculation of animal cell inside the media in the culture flask, you have to keep your culture flask in horizontal position. Understand? So that it will get a suitable environment for the growth and multiplication of animal cell. you know CO2 incubator, it provides sufficient amount of carbon dioxide, sufficient amount of relative humidity, and it also maintain the temperature. It also maintain the temperature. So just see in the diagram, it is a CO2 incubator. You can adjust or calibrate and maintain the temperature, relative humidity, CO2 in optimum concentration. or optimum quantity so that it make a favorable condition for the growth and multiplication of animal cell line. So one of the most important instrument that is going to utilize in animal cell culture is CO2 incubator. Before that, you do all the activity inside the laminar airflow that attach with HEPA filter. Okay, HEPA filter, it is a special type of filter that allow only purify air inside the laminar air hood. Okay, next instrument, you know, that is centrifuge. So what do you mean by centrifuge? It will rotate revolution per minute. As per revolution per minute, you will fix the RPM, and then you collect the whatever the compound you want to collect, or you want to isolate the cell. Then in the Ockridge tube or Appendot tube, you put the animal cell, and then you put inside the rotor. Okay, inside the centrifuge, and that centrifuge must be refrigerated. Why it should be refrigerated? Because animal cell culture are highly sophisticated, delicate, and it may be affected due to environmental temperature and the heat generated due to rotation of the motor of the centrifuge. So it can affect the physical environment, and it will create a disturbance, and it can degrade the animal cell. So that we prefer to use the refrigerated centrifuge. Refrigerated means cooling system must attach. Whatever the heat released during rotation of the rotor by the motors, it will absorb through the cooling system, and your cell, what you are going to isolate the animal cell, it will not going to affect due to heat released due to rotation of the motor or rotors of the centrifuge. So we prefer a refrigerated centrifuge instead of normal centrifuge. Okay, next instrument, that is inverted microscope. Okay, you try to know what is inverted microscope. Means you observe the illumination system at the bottom, and if you grow the animal cell line, if you grow the animal cell line, it will grow from the bottom side. If you see figure number 2, that is culture flask, you put the culture flask at horizontal position after inoculating the animal cell inside the culture flask, and it will start growing on the horizontal surface of that culture flask. So it will attach to the bottom, not on the top of the liquid media layer. You know, in case of animal cell culture, generally the animal cell line will multiply from the bottom, from the bottom. So that is very very important. So you have to observe the animal cell growth pattern from the bottom, not from the top. So there must be elimination system from the bottom line in case of microscope. So we prefer the inverted microscope. We prefer the inverted microscope for proper study of animal cell culture. And if you are using the normal microscope, you cannot observe properly the growth pattern of the animal cell. So this is very very important point for your examination point of view. For any competitive exam, generally ask why we use the inverted microscope instead of normal microscope for the growth of microbial cell culture. Sorry, for the animal cell culture, because growth pattern you can observe from the bottom layer of the cultural flask in case of animal cell culture. But if you observe from the top through the normal microscope, you cannot observe properly because there is a liquid media will present on the surface of the animal cell culture, and you cannot observe properly in that situation. So the illumination system must be at the bottom for proper study of the growth pattern of the inverted microscope. Through the inverted microscope for the study of animal cell culture study. So next question may be asked in so many competitive exam, that is refrigerated centrifuge. If you want to maintain your animal cell culture properly for the further study during isolation and collection of the animal cell, you have to use the refrigerated system, because if it will affected by heat, it will degrade or it will be deteriorate. So you prefer the refrigerated centrifuge instead of normal centrifuge. Okay, I have already discussed a CO2 incubator. Why we prefer the CO2 incubator? It provides the favorable environment for the growth and multiplication of the animal cell culture line. Why? What are the different favorable environment? It provides the proper relative humidity, it provides the proper concentration of carbon dioxide, and it provides the proper optimum temperature. I am talking about optimum, not maximum, not minimum. Whatever the temperature required, you have to adjust the CO2 incubator, and it should be free from any viruses, any microbes like bacteria, fungus inside the CO2 incubator chamber. So you will be confident that your experiment will be successful when you are growing your animal cell line inside the animal cell culture flask after putting inside the CO2 incubator. So this type of question may be asked: why we prefer the CO2 incubator, why we prefer the refrigerated centrifuge, why we prefer the inverted microscope. Such type of question may be asked in any competitive exam or your coming board exam. Okay, so this thing you have to maintain in for the proper study of animal cell culture. Uh, as you know, if you compare the animal cell line, you have to observe from the bottom line. That's why we prefer the inverted microscope. Thank you and have a nice day.